Hello, everyone, and welcome to the second webinar of the third series of webinar focusing on the use of proteomics in clinical applications. So for your information, this webinar is recorded and you can ask your question to our speaker through the Q&A box. So this webinar is organized by the Education and Training Committee, which is one of the initiatives from International HUPO Scientific Organization, focusing on disseminating knowledge from experienced scientists to the whole MS community. You can follow our activities on our webpage and on the HUPO website. Talking about the events we organize, you can see that we have few talks already scheduled. If you want to take a few seconds to screenshot this table, you can see all the events that will come in the following weeks. Also, we are leaving in free access all the records of the past webinars that you can watch on YouTube. You can go on the HUPO channel to watch them. And we also write summaries of those webinars, and the first published editorial opinion is available on the website of Journal of Proteome Research. So focusing on today, Giuseppe Palmisano and myself, Blandine Chazarin, we are delighted to welcome Professor Isabelle Fournier, who will present the innovative technology named SpiderMass. So to say in few words, Professor Fournier is distinguished professor at the University of Lille in France, and she is co-director of PRISM Laboratory. She is a bioanalytical chemist specialized in clinical mass spectrometry and proteomics. She started by working on fundamental MALDI during her PhD at University Pierre and Marie Curie in Paris, before moving to a postdoctoral position in the group of Professor Michael Carras. In 2002, she established her group at University of Lille in France, developing MALDI MS imaging. She has pioneered the field by developing strategies for imaging on FFPE tissue and protein detection, and the development of tagged probes for targeted MS imaging. Since 2009, she is a full professor at University of Lille and has a junior position at the University Institute in France. And since 2012, she's invested in the implementation of a specially resolved proteomics by MALDI MSI to be used in oncology. Now she is massively working on in vivo MS to guide surgery and provide intraoperative analysis, which lead to the development of the SpiderMass technology that she will present today. So now it's time for me to leave the floor to Professor Isabelle Fournier for her presentation named The Amazing SpiderMass, a game-changing technology for precision surgery through MS-based digital twins.
Many thanks, Blandine, for this very kind introduction, and thanks, Blandine and Giuseppe, for hosting me today for this virtual HUPO seminar. So let's first share my screen, and just can you confirm that you can well see my presentation?
Yes, we can.
Yes, we can see.
So let's start. So today I would like to focus on, as Blandine was mentioning, a recent technology that we are developing in the lab that we have coined with the name SpiderMass, and with which we like to address the problematic of guiding the surgery. So maybe let's start and let's come back on the very basic of this project. This project came from a tight collaboration we had with Dr. Leblanc, who is a famous surgeon and is head of the gynecology department in the comprehensive cancer in Lille. And Dr. Leblanc was mentioning to us that despite many developments in the surgery, in particular in the field of robotics with the Da Vinci system, they still struggle to be sure that they can reach the optimized cancer surgery, and in particular in their case, that means being able to be sure that they have removed all cancer cells, because it's well known that in case that the cancer cells are left, they will be relapse of the patient. Something that is very important is also to realize that in the arsenals of cancer therapy, surgery still remains one of the first frontline treatment, even that most of patients go for chemotherapies and some of patient radiotherapies. Surgery is still something at the front line, and there are more and more patients to be predicting to have a cancer and need a surgery by 2030. So there is a huge importance to be sure that we can offer to this patient surgery of quality, and considering that the patient, their outcome will be driven by the quality of the surgery. Currently, the gold standard for the surgeries is still based on the pathology examination, also called intraoperative pathology examination. This means that during the surgery, the surgeons will take some samples, take some biopsies inside the patient at the best of his knowledge, because he can only rely on his expertise and on his eyes. And he will give these samples to the pathologist that will try in the minimum of time possible to perform a diagnosis. In the short frame time that is available during the surgery, they can only go for normal conventional staining and conventional histological examination, which means that there is a known and quite important error rate by comparison to what they can do during the post-surgical exam. In particular way, they can access more refined technologies such as immunohistochemistry or in situ hybridization. And because of that, there is a lot of errors that are obtained on the sample diagnostic. And this means that first, generally, they still are cancer cells in the patient, and the margins for the excision tissue are not free of cancer cell. And that means that sometimes for some pathology, up to third of the patient need to be reoperated after the results coming from the post-surgical examination by the pathologist. So all of this, the fact that this intraoperative gold standard is not that great, has a lot of consequences to the patient, because first, if you leave the cancer cells, as I was mentioning, you will have important relapse rates, but also the surgeon might miss the presence of metastasis, which will also decrease definitively the outcome of the patient, and decrease the overall survival, but also impair the patient quality, because in the case the surgeon might have some doubts, the decision could be to remove more tissues, but then you will decrease the quality of life of the patient. For all these reasons, we have decided to try to see whether we could develop something that would be based on mass spectrometry to help the surgeon directly during the surgery. And the fact was that there was no direct technology that could be used as so for this purpose, and therefore we had to think about new technology to match this goal. Maybe you can think that it's a bit crazy to think bringing a mass spectrometer into a surgery room. Yet, since more than 60 years, we are also sending mass spectrometer in space, especially with the spatial mission. Whatever these mass spectrometers are, starting from quadrupole to 2D or 3D ion traps and time of flight, and there are now even development of orbit trap for space missions. So considering that you can develop an instrument that's smaller and sufficiently versatile to match the need of a special mission, that it's not that impossible to think of, moving a mass spectrometer to the surgery room. So even if it seems a bit crazy at the beginning to think installing a mass spectrometer inside the surgery room, that's not something impossible, and that is really the challenge we wanted to tackle with this project. And as you can see on this drawing that I have made, we have even a quite high spectral resolution mass spectrometer like the Explorit, which are relatively already small inside, even if they are not, movable or from one room to another. Still, they're already quite small in size and showing very high performances. Regarding the application to the surgery room, this is something that was rendered possible at the beginning. If you look back to the map of development of mass spectrometry, that is something that is directly coming from the development of the so-called ambient ionization mass spectrometry sources, which started with the APC, atmospheric pressure chemical ionization, and then also the desorption electrospray ionization. I think DESI was a great step in the development of ambient ionization mass spectrometry. And but nevertheless, even if DESI is great, DESI was not appropriate for in vivo analysis of patients. DESI can perform ex vivo analysis at the surgery room intraoperatively, but is not fit for in vivo analysis. Therefore, there were other technologies that was really developed for the purpose of intraoperative analysis, but also not only intraoperative analysis, but intraoperative analysis performed in vivo, so directly in situ to the patient. The technology that were developed based on mass spectrometry for in vivo application are regrouped here. And the first one that was ever introduced was the so-called iKnife or RAMS technology. Then followed our technology, the spider mass, and there is also the mass spec pen and the PAL technology that came later on. To give you some background about this technology before switching to the spider mass itself, the first technology, that's the iKnife technology, was a vision where they tried to couple a surgical instrument like a surgical knife or surgical forceps to a mass spectrometer, and they were collecting the smoke emitted by the system, as you can see from this video, to transfer them sent to a tubing, to a transfer line, to a mass spectrometer that would analyze the smokes in real time. And you probably know that this system, has led to many publications and has shown to enable follow-up during surgery. Yet, one of the only drawbacks of this technology is the fact that you only get the information while you are cutting through the tissue. And in our case, indeed, that was something that the surgeon were really wanted to avoid. They would prefer to have something less invasive and have the information before deciding what tissue to be removed. Another technology, to give you a bit of background, that was developed a bit more recently is the mass spec pen developed by Livia Berlin. And Livia, she is at the University of Austin in Texas. And the mass spec pen is based on, it's a pen where you can generate with a simple pressure of the foot a small droplet of water that will go into contact with the tissue, enables to extract the markers at the surface of the tissue, and then the droplet, as you can see of the video, will be sucked back and bring back through this transfer line to the mass spectrometer that is equipped with a specific ion source and interface for real-time analysis. And so the mass spec pen over the iKnife has the advantage to be very limited invasiveness because it's only an extraction with water, but has the limitation that the system needs to be put into tight contact with the tissue to be analyzed. On our side, when we decided to tackle this challenge of mass spectrometry analysis for surgery, we decided to try to get we had a certain number of things we wanted to address with the surgeon. The first thing was to have a versatile system that we could use both intraoperatively, both ex vivo, but also in vivo in situ in the patient, but also to have a system that we could adapt to perform ex vivo and also help the surgeon post-surgery. The second thing we wanted with the system was to be able to be of very limited invasiveness so that the surgeon could first analyze and then only after decide what tissue to be removed. And then the last point that was very important for us was to be able to perform an analysis without the need to put the probe into contact with the tissue to avoid cross-contamination between the different part of the tissue that we would analyze. And this is the reason why, for the spider mask technology, we decided to go for a system that would be based on using a laser. And the basic concept beside the spider mask technology, that is based on what we call water-assisted laser desorption ionization mass spectrometry, or WALDMS, is to try to perform or to process that is similar to MALDI, except that in the case of MALDI, you have to add an organic matrix in your experiment. And the idea with the spider mask was to use the endogenous water of the biological tissue as a MALDI matrix. And therefore, we have developed a system where, with a laser emitting in the mid-infrared at 9.4 micrometer, we will perform resonant excitation of the endogenous water molecule. We still use, as for MALDI, a nanoseconds pulse duration laser. And we have managed also to develop, which was a technological breakthrough, but we managed to develop a laser that is fiber, meaning that at the exit of the laser, the light is injected inside an optical fiber, at the exit of which we have a handheld that you can see here on this picture, where we have the focusing lens to be sure to control the spot size of the analysis. And as you will see, thanks to this laser, we have a system that is of very limited invasiveness, where, thanks to the resonant excitation of the water molecule inside the biological tissue, we can get a small desorption ionization and production of an aerosol. But as you can see, the damages to the biological tissues are really minimums, and it's only leaving a white trace that will be disappearing within seconds or minutes after the experiments. And then this aerosol is sucked back to the mass spectrometer thanks to a transfer line that is this plastic Tygon tubing that you can see here on the video. And then the mass spectrometer will operate in real time to generate the mass spectra. And for sure, we have also a last part in the system that is a data processing, which is based on artificial intelligence, but I will come back later on this aspect. We have a different prototype in the lab. One prototype that we have is our spider mask. You can see the laser here is connected to a QTOF from Waters Instruments for the prototype of the commercial RAMS instrument. But we have also developed our own interface to connect the transfer line to the mass spectrometer. We have developed two interfaces that are protected by buttons. One that you can see here, that is for connection to a QTOF from Bruker, and one that you can see here, which has a bit different process that is set up for connection to a Orbitrap instrument, and in that case to a Q-Exactive instrument. Just to show you some example of spectra that we can obtain directly in situ with the spider mask. As you can see, we mainly observe from tissue signal from metabolites and lipids. And here you have a nice example that we perform in collaboration with Pierre Dermocosmetic Laboratories, where we were following the evolution of the metabolites and lipid profiles from the skin of patients with seborrheic dermatitis before and after the treatment with a formulation, a new formulation from Pierre laboratory. And this is a formulation with a new antifungus active product. And as you can see from this spectra, we can very see the differences before and after the application of the treatment. And in particular, we see after application of the treatment of a decrease of the metabolite and lipid signal, which correspond to pro-inflammatory molecules. What is very interesting for us, because we wanted also to have an idea of the quantification performances of the spider mask system. So we have been comparing for the molecule we could find in common, the spider mass, that is a shotgun approach where we don't use any separative technologies or affinated technologies to a conventional LCMS metabolite analysis that was performed by a company. And as you can see from this graph, from I have only plotted a part of the metabolites and lipids that were identified in this experiment, but as you can see, we have a very good correlation between the LCMS analysis and the spider mass experiment for the quantification. We have also been checking whether we could analyze protein using the spider mass system. And indeed, spider mass can get access also to protein signals. Very interestingly, what we observe by analyzing protein standard is that if we analyze protein standards with spider mass, we obtain mass spectra which are very similar to electrospray spectra, meaning that unless what we have in MALDI, in the case of the spider mass, we have highly charged state proteins. From this basis, we were trying to see whether we could get some signals of proteins directly from tissue. And you have here some example where we recorded spectra from beef liver model tissues, where you have here in the lower mass the monocharge signal corresponding to the metabolites and the lipids, but you can see here that we also have some signals with much higher charge states that correspond to proteins. So far, the proteins that we observe are only major proteins of the tissue, and in particular, in this case, we did perform a top-down in the Achilles cell of the Hodgkin trap on this signal that we were observing in the spectra, and we identify major proteins like the hemoglobin of the tissue. But still, we know that we can perform top-down approach from this spider mass experiment. And we did also started to check from tissues that were performed directly from dog patient tissues that were collected in the surgery room. And again, even if we still have a lot of lipid and metabolite signals, we can start to see some signals corresponding to higher charge states, which are definitively attributed to proteins. And these signals were observed both ex vivo on the bench during the surgery or directly from the spectra recorded on the dog patient. So proteomics is not perfectly there with the spider mass, so we have some this preliminary data, and we are now moving to try to improve the signal of the protein in the spectra by comparison to the lipids and metabolites. So we know the unique difficulty that we will have to challenge is to get rid of the abundant lipid and metabolite signals, and one of the possibilities that we could have would be to use higher mobility like FAMS. For sure, targeting or forcing application for the surgeon, we cannot ask the surgeon to look into mass spectra. Therefore, we really need to rely on artificial intelligence and on particular onto machine learning experiment. So what we are generally doing is that if we want to address clinical problematics or surgery, we need first, before implementing the system directly in the surgery room, to receive some sample from the pathology lab, which has a good annotation for which the pathologists are sure of the annotation. Then on this, we will record spider mass spectra, give the spider mass spectra a class corresponding to the annotation by the pathologist, and based on that, we will create classification model. Here you have an example of classification model for sarcoma using conventional machine learning and in particular linear discriminant analysis. And whenever this classification model are built, then we can, deploy the spider mass, and the spider mass can be used directly in the surgery. So here you have an example of a video that was conducted in the vet surgery room on dog patients with sarcoma. And the idea is to get something that is simple to read by the surgeon. For example, in that case, a color code that tells the surgeon whether the tissue is cancer or non-cancer, or what is even the subtype of the cancer that they are looking to. We already have a first in man that was conducted together with Perfab Dermocosmetic for patients with skin pathologies. We were in particular interested in the inflammatory disease of the skin. So we received, they have received in Hôtel Dieu in Toulouse patients with different type of pathology that ranging from atopic dermatitis to acne or psoriasis and pathology of that type. And as you can see here on this picture, so the patients were analyzed either on the forearm and even on the face. And this is a clinical register, clinical trial. So that's confirmed that the system is not dangerous and very microinvasive and can be really used in vivo. And as you can see here on the right from this video, we can even perform automated acquisition from some area of the skin here on the forehead of this patient, and luckily the hair are not absorbing at the wavelength that we are using, so the hair are not burning and stay there. And this experiment that we conduct for the skin diseases are very informative for the people to better understand the pathophysiological mechanism associated to these diseases, and also for the laboratory to develop the new product or to better optimize the treatment for the patient. What we have recently done was also to shift our Spider-Man system from a manual system where you could take in the hand the laser probe and analyze whatever tissue you want, to a system that would be more automated and that could be able also to perform imaging. So to move to Spider-Man imaging, to this end, we acquired this small robotic arm that is small but has a very high precision of accuracy of positioning, and we connected the probe of our laser onto this robotic arm. So you can see here there is the handheld with the focusing lens of the spider mask. Additionally, inside this part of the robotic arm, we added a distance sensor to be able to measure the distance to the sample, adjust the laser focusing, and also get the topography of the tissue that we would analyze. We have also been developing a new laser system that is more compact for the surgery room and equipped with a jacketed optical fiber that has plug-and-play connection. These are examples of some images that we perform from histological tissue section of esophageal cancer. This is a comparison here of the same tissues or consecutive tissues analyzed conventionally by multi-mass spectrometry imaging with the North-Harman matrix for lipids, and here the spider mass imaging. And as you can see from these different images, except from the resolution, because so far the spider mass, we have a spot size of 500 micrometer, which match the need of the surgeon, and we can go down to 250 micron by performing oversampling, and the multi-imaging can go much down this value and have much better spatial resolution. In that case, we use 50 micrometron spatial resolution, but except from the spatial resolution, we have the exactly same distribution of the markers for the multi-imaging and the spider mass imaging. As I was mentioning, our system enables to measure the topographic of the object that we are measuring. So here we have our favorite patient, that's Bob the Sponge, and you can see that we can get the topography of the object. And here you have an example where we were testing this on the figure of postdoc of the lab, where we did this pitch of imaging on the tip of the finger, where we get, thanks to the distance sensor, the topography of the finger, and then we could plot back the molecular imaging data from the spider mass imaging onto this topographic map. So now we are also able to produce molecular topographic imaging, and that is the base that we want to use to offer and propose digital twins to the surgeon for the surgery. We have also worked on the data processing because, as I was mentioning, data processing is very important because this is makes the data interpretable and usable for the end user. That's the surgeon or the pathologist. So we have been looking and trying to improve the quality of the classification models by changing and testing different type of machine learning algorithm, and then also moving to deep learning models. And this is an example where we were testing this development on a cohort of 50 prospective tissue, fresh frozen prospective tissue of patients with glioblastoma. And we also had 50 retrospective cohort tissue of FFP tissue of other patients with glioblastoma. And we were using the spider mass spectra from all this tissue to try to create a classification model that would be strong enough, because here we have a total cohort of 100 patients. And as you can see, what was interesting is that we have found out that we could perform spider mass spectra both from fresh frozen tissue but as well from FFPE tissue. That was the first thing that was very interesting for us. The second thing that was very interesting is that we could the spectra between fresh frozen and FFPE tissue were so sufficiently close in the molecular profile that we could combine both type of spectra to create in a single classification model, in a single training and learning experiment. And from this mix of fresh frozen and FFPE tissue, we could create a classification model which could reach 92% of good classification, and that we could test the performance of this model on blind tissue of glioblastoma. And you could see that from the 40 tissues and 40 predictions that we made in blind after training the model, we only had 2 errors for 2 regions that were necrotic regions. That was the class of annotated sample that was the less well represented in our model. Something that we were also interested in was to try to see whether we could gain from the tissue information about the immune cells, because in cancer tissues, the microenvironment and the immune cell in the microenvironment are fundamental, especially for the outcome of the patient, the response to the treatment, and the overall survival. So for this experiment, what we have been doing is that we have isolated immune cell from blood samples of patients. The cells were isolated by FACS. They were FACS sorted, so we could isolate the different types of immune cells. And then we were analyzing all these different immune cell subtypes with the spider mass individually. And then we were looking to the classification model to see whether these immune cells were showing different molecular profiles. And as you can see from this result, the immune cells have different profiles in the spider mass than the cancer cell lines that you can see there, represented there. But if you only look to the immune cells themselves, you can also see that we can easily discriminate between the different types of immune cells, the macrophages versus the lymphocytes, and we can even subtype the lymphocytes according to their different subtypes, such as LTCD4 or LTCD8 or NK. And we could even also discriminate between the M1 or the M2 types of macrophages. So that is very interesting. But the second step of this story was not just to analyze the immune cell alone, that was to also try to identify what was the marker that were specific to these different immune cells. And here you have some examples of lipid markers that were identified to be specific either to M1 or M2 type macrophages, and also the markers that were identified to be specific for the lymphocyte. And very interestingly, what we could see is that if we were looking for this marker in the different tissue of the patient that we had analyzed previously, we can really see that the abundance of this marker is very different whether you analyze healthy or cancer tissue. Something that we also did, because we know that we will not target the single cell analysis. I mean, single cell proteomics is something very interesting nowadays. But in our case, we know that we won't target single cell because we will never ask the surgeon to perform single cell analysis nor to try to cut to single cell. Therefore, we have decided to take another paradigm for the surgery, that is, we will keep a quite large laser spot, but what we will do is try to repredict what is the cell ratio in this large analytical spot. And this is what we have done thanks to a deep learning algorithm pipeline that we have developed here using the LightGBM algorithm from Google, where we could, from a mass spectrum, thanks to the learning with the single cell line isolated, then from the tissue be able at a specific spot to predict the ratio of the different cells in the spot. And you have an example where this strategy was applied on a spider mass image of glioblastoma, and then we can predict where are the different ratio of the different cell types within this image. And that's very interesting because that means that we can also now find directly in the spider mass and potentially in the short future transfer this also to the patient in vivo during the surgery to predict the M1 and M2 and the lymphocyte, and also use this ratio of macrophages M1, M2, or the ratio between macrophages and lymphocyte to be able to predict the overall survival of the patient already from the initial time point of the surgery, because it is well known that the ratio between M1 and M2 or macrophage and lymphocyte are predicting the overall survival. And what you see there is this applied to our glioblastoma patient cohort. And we can really show that the score that we obtain, thanks to the prediction based on the spider mass image, are matching with the overall survival of the patient that were observed from the cohort we had. We can use the similar algorithm also to address the problematic of margin delineation. That is also an important and paramount problematic for the surgeon during the operation. And as you can see there, we have been simulating a process of margin delineation on a cancer tissue. In that case, this was a cohort of esophageal patient, where from histological here tissue section of esophageal cancer, we were analyzing, performing some row of analysis with the spider mass. So we were shifting the spider mass analysis from one consecutive point to another, and then trying to see whether we could get a classification of these different areas of the tissue as cancer, non-cancer. But we have also very fastly realized that the classification as cancer or non-cancer is not sufficient to properly address the problematic of margin delineation, because you have a gray area where you not only have cancer or only healthy cells, but you have a mixture of them. And thanks to the LightGBM algorithm that I presented just before, now we can really go also for percentage of cell ratio between normal and cancer cell. And we can really say that, okay, there is only cancer cell. Here there is less than 25% cancer cells. So we can really decide what the surgeon needs as information to be sure what he will decide whether to remove or not this tissue. And as you can see, we can have a gradient of color corresponding to different mixtures of cell population. And for sure, thanks to the mass spectrometry, we can also identify the markers which are specific from the LC, the cancer region, or the gray area where we have a mixture of LC and cancer region. As you can see, we have been also aggregating our spider mass data and learning to the clinical data of the patient that were collected by at the hospital. And what was interesting and logical for the esophageal cancer is that we have found that the three main predictive parameters for overall survival in esophageal cancer is the age, the tissue type, because there is an aggressive subtype of esophageal cancer that is the poorly cohesive carcinoma, which is a more aggressive subtype generally observed in younger patients. And that's definitely this patient which has a very poor overall survival. And there is also a relation with the staging of the cancer based on the presence of tumor metastasis and also the positive or negative lymph nodes. And whether we aggregate our spider mass data to all this clinical data, very interestingly, what we found in particular for the subtype of the overall survival that is due to the subtype of the tissue is that the lipids we identify with the spider mass are the parameters in the model which give the more explanation for the survival of the patient, and that was something we were really happy to see. And also, if we look to the overall survival based on the esophageal cancer staging, we have also found that some of the lipids that we have identified with the spider mass were among the parameters giving the more significance to the overall survival models. We get also interested in the esophageal cancer to the bacterial problematic because there are a lot of bacteria in the gut. And this is something to be considered because it's well known that there is a dysbiose of bacteria in the patient with pathology, whether they are inflammatory pathologies or cancer pathologies. So we are analyzing a large population of different bacteria for which we collected the spectra. We have analyzed 52 bacterial strains, and you can see here the representation of these bacterial strains. And for example, you can see that in our tissues, the representation of this bacterial strain is very different. And from this bacterial strand and the molecular signature on this bacterial strand, we have been trying to create a classification model to be able to recognize from the patient tissue the presence or not of this bacterial in the tissue. And we were able, on this 52 bacterial, to get classification model that could reach up to 96% accuracy. We have then applied this as previously showed to the images obtained with the spider mass from the tissues of the patient with esophageal cancer. And here you have a distribution of some of the bacteria. So you can see that for this tissue, these two bacteria, they are absolutely not present, but there is a huge, for example, presence of Helicobacter pylori, which is known to be a factor of cancer onset in esophageal cancer, but it's not the only bacteria that is detected in this patient. And this is the different bacteria and the abundance that we detected thanks to the machine learning inside the tissue of the patient with esophageal cancer. Again, Helicobacter pylori is the more prevalent, but we also observe other types of bacteria. Interestingly, the bacteria types is also different according to the subtype of esophageal cancer. And definitively, if we compare the poorly cohesive carcinoma aggressive subtype, we can see different populations. So, for example, some bacteria are totally missing in this aggressive subtype of cancer, where other bacterial populations are much less present than in the more conventional adenocarcinoma of esophageal cancer. And as you can see, we have also confirmed the presence of different markers with different expression according to the subtype of the esophageal cancer and the type of bacteria we were looking for. Evidently, as you have understood, all of this is based on machine learning and deep learning prediction. And therefore, to be sure what we found, we always want to perform a validation of what we find. To perform a validation, we like to rely on MALDI immunohistochemistry or MALDI imaging with immunohistochemistry, which is something that we developed already a long time ago in the lab. The concept of this, I guess you probably already know MALDI-HI, but it's to modify an antibody to be able to graft on it a tag that bears a reporter that can be detected, that can be released by UV light and then detected when performing MALDI mass spectrometry imaging. We have recently, it's a very old project for us, but we have been recently redesigning our probe to be compatible with chemistry, which give us much more flexibility to create and modify any antibody that we have in the lab or that we need to modify. So we have decided to still keep a photocleavable moiety to perform the release of the reporter. But this is added by click chemistry that's more flexible. This is done what we are performing. We have also shift from a MS to a MS reporter group that is much smaller in size. And before we were using peptides, now we are using small molecules which are derivative of arginines. And with this derivative of arginines, we have already been able to synthesize 10 different tags. And we can perform the immunohistochemistry sequence with mass spectrometry imaging performed either on the Rapiflex or the TimStofFlex MS instrument. And this is a validation of the margins that we did perform and we did predict with the machine learning and the SpiderMass. Then we have validated that the margins that we found with SpiderMass are correct by targeting markers of cancer cells. Here it was a Ki-67 confirmation with MALDI EHC mass spectrometry imaging. As I was mentioning, we have now 10 tags which are ready. So as you can see here as an example performed from a FFPE tissue section of a breast cancer, we can now address at least 10 different markers on the same tissue section. Here we were looking in the cancer cell tissue to markers of immune cell as well as immune checkpoints markers, and you can see that we have very nice imaging performed in the same immunohistochemistry experiment and then revealed by mass spectrometry, multi-mass spectrometry imaging. And our plan is to use this 10-plex to be able to confirm the prediction that we have on the bacteria by targeting with modified antibody and multi-10-plex immunohistochemistry the presence of the bacteria in the esophageal cancer tissue. We are now, as I was mentioning, the aim is also to offer digital twins to the surgeon. So we are now trying to use the interpretation by the machine learning of the multi-imaging data and combine this with the topography measurement of the tissue of the patient. The final goal of that would be to merge this with the image that can be obtained from the camera that the surgeon will have in the surgery. And here you have an example where we were analyzing a transgenic mice model that's mimic triple negative breast cancer. And as you can see, we can perform direct imaging. This is a post-mortem experiment where we were imaging the mammary gland of these mice. And you can see here the reconstruction and interpretation of the SpiderMass data recording in real time. And at the end of the experiment, we obtain this type of maps where you have both the molecular interpreted data and the topography. And from this base, from this digital twin, the surgeon can decide, he knows where are the tumor cells, where are the non-tumor cells, and he can or she can decide where to cut, what the tissue to be cut and what the tissue to be left in the patient. And for sure, we can also give a lot of different interpretation to this molecular topographic map or digital twins, included segmentation analysis, as you can see there, and TSN representation to find in blind the different region in the molecular map. But we can also start to include the search of the bacteria within this digital twins, also to offer to the surgeon the possibility, if needed, to monitor the presence of these different bacteria, also as a means to be able to foresee digital things predicting the outcome and the overall survival of the patient. Now we are moving to imaging. I mean, we want to perform the imaging at a time that is compatible with the time of the surgery. So we are now moving from a spot-to-spot conventional mode of performing imaging to a continuous mode where instead of shooting acquiring the spectrum, moving to the next spot, shooting, etc. We are now continuously moving the laser probe thanks to the robotic arm. And you have here some example of image that we were able to generate at different spatial resolution, ranging from 240 micrometers down to 60 micrometers. And what is interesting is that the image at 240 micrometers was acquired in 6 minutes. And considering that we are even monitoring area outside the tissue section, that means that the area of the tissue section itself was imaged in less than 5 minutes. And that means that we are now compatible with the time that the surgeon can afford to perform image during the surgery. So we would like also to offer the surgeon to have automatic acquisition on certain suspect area and really creation of image digital twins from this suspect area that are targeted. And as you can see here, we have a very nice segmentation. So we can, after we get the image, also offer different types of interpretation, including image segmentation. And the really big challenge for us now will be to miniaturize our spider mask system. And we also work with colleagues at INRIA to be able to include the spider mask into a soft robotic silicone system. So the idea would be to miniaturize enough the laser fiber as the transfer tube, so we could insert that inside this small robotic robot that can be controlled from a computer, and that is moving thanks to application of different pressure inside the chamber of the silicone system. So this is our next challenge, is to integrate the spider mask into this robotic system and also include the robot to endoscope, so we can also target mini-invasive surgery and offer imaging and interpretation for the surgeons directly for endoscopy, laparoscopy, or coloscopy. The very last thing I wanted to talk very briefly is that we are also now trying to bridge a lot the gap between multi-mass spectrometry imaging and proteomics. And our idea with this is to see whether we could fastly perform images. Here, in this example, the images are generated with conventional multi-mass spectrometry imaging, but in the future, we would like to replace that with spider-mass imaging. And from the simple image of the lipids would be to directly associate the protein pathways, also to be able to get information for the therapeutics. So briefly, the idea of this project is to get the lipids imaging, get the protein imaging, then get the lipids identification and signaled pathways on one side. On the other hand, get bispatial proteomics, the signaled pathways for the proteins, and then use machine learning to associate the lipids and the proteins together. This is where we have been using the glioblastoma as an example to make a proof of concept of this concept that we have called right proteomics, and that was very recently published in Molecular and Cellular Proteomics. You can see from the image segmentation, the global segmentation of the image on this small cohort of patients with glioblastoma, that the imaging segmentation give a separation of the tissue according to the survival of the patient. The red circle group being the patient with the lowest survival, and the green being the patient with the longest survival. And from that, thanks to the machine learning, we can directly associate the protein pathways to different tissues and different groups, thanks to the spatial proteomic. After doing this learning session, we can then deploy the system in blind, okay? And that means that here you have the blind application where automatically for an unknown sample, we can get its classification in poor or good overall survival. So we can know the prognosis from the patient and then automatically rolled out the protein pathways associated to the tissue. And this is very interesting in different situations, such as breast cancer, where we have a huge heterogeneity, clonal heterogeneity, is, as it is highlighted there, from multi-mass spectrometry imaging performed in the bottom up from the tryptic peptides and after the segmentation of the tissues. And we know that there is a lot of patients with resistant to treatment and still patients that cannot that will die from breast cancer. And because of that, and because we can follow also and monitor in the longitudinal view all these patients with different patient subtypes receiving different therapy, thanks to the dry proteomic concept, what we now also target is to use this concept on organoids from patients. And see, here you have an example where we were monitoring the organoids with the multi-imaging of lipid that very fastly done. And then from that, we automatically retrieve the proteins pathways. And then we can also foresee the adapting the treatment to this patient. And in the case of this organoids, what we found is that the patient that was given to the patient is apparently not the best treatment, because we finally found that we had better result with using a different treatment that is sunitinib by comparison to the paclitaxel that was indeed given to the patient. And with that, we will now move to do the same thing with the spider mass imaging. And maybe one day we could offer that to be performed in vivo during the surgery as well, already trying to foresee what's the best treatment to give to a patient. I will probably stop there. We are also developing mass spectrometry imaging based on LFQ proteomics, but that's a different story, and I think I was already quite long. So if you have questions, I can answer during the Q&A session, but I will probably skip this part. And I can just show you that, we can also perform images from large-scale proteomics data, as you can see from there, but just ask questions if you want to know more about this part. And with that, I want to thank all the people that were involved in this project, in particular the students, the PhD, and also the funding. And if one day you want to buy a spider mask, we have now a small spin-off company that is trying to industrialize the technologies. And really, thank you for your attention.
Thanks a lot, Professor Isabelle. Was very enlightening presentation. Since the beginning of the development until the real recent application. So I will start actually seeing here if we have some question. We have one question actually from Justina.
Do you prefer I keep the PowerPoint for the Q&A session in case people want maybe to see, or go back on to certain? Okay, sure.
I think it's a great idea. So in case you need to move on the slide. So Justina has actually two questions. The first one is, what are the limitations of laser ablation ionization in complex tissue environments?
We don't, that's a shotgun approach. We are not using any separation technology. So for sure, we won't see as much as we can see by conventional large-scale proteomics approach. We are not like identifying these 10,000, 10,000 proteins or lipids, so for sure. But yet we already see thousands of different signals, thousands of different molecules. And as you can see from the example I have given, that's sufficient at least to be able to, because we don't only process a few markers. What we do with the machine learning is we take the global molecular signature and all the peaks with all the intensity for the learning. We also try to always go for LSMS and identification and cross-validation of what we observe in the signature. But for the application in the surgery, we really want to use the signature themselves. As you can see, I think they're already quite specific because we're able to find the presence of specific peaks to the different immune cells or the different bacteria within this profile. So I think we already have quite a lot of signals and information. we can still improve the thing. One idea that something that we would love to try, but so far it was not possible for instrumental reason in the lab because the instrument is already used for large-scale proteomics. Something that would be interesting for us would be probably to couple the spider mass to the timsTOF and benefit from the TIMS technology.
Yeah, mobility, yes.
But yeah.
Great. I think you already partially answered the other question. So it was what improvements are needed in resolution to identify more complex markers. I believe in terms of identifying more features that allow you to separate better the
No,
different.
that's a good question. We are doing a lot of the work that was achieved here was performed on QTOF instrument, and we have a good resolution, but it's not extra good resolution. I mean, we will never. We are not reaching like 100,000. And definitively, this is something that we observed when we were working on the Orbitrap instrument, that we see more features. We also have more stability of the calibration of the mass to charge. So yeah, in this respect, the Orbitrap technology is interesting, despite it's a bit slower, as you all know. So, but yeah, we plan now to try to develop a bit more the spider mass on the Orbitrap instrument. So we use the Q-Exactive because now people they have all moved to the tribrid that we recently acquired. So we have one spare QX. So we will probably do more experiment on the QS because definitively having more spectral resolution would also help seeing and being even more specific in the interpretation. That for sure.
Keep going on the question here. You mentioned this development of the stiff robots, right? This robotic arm, this new version that actually allows you to go less invasively into the tissue. Do you think there will be an improvement also in the algorithms that actually controls these robots in order to better map the tissue?
So maybe I can show back this. Yeah, you have the video there. So yeah, actually we are the PhD student working on this robot. And as you can see, it has a very complex numbers on the computer screen. So he's using a very, very interesting simulation software that's called SOFA, that was developed by some colleagues specialized in robotics in Lille. And definitively is also, we have some rooms for improving the controlling the movement of the soft robot. So for the stiff robotic arm that we use in the lab, for this we have a very precise control because that stiff part of the robot that can be moved. So for this we can reach 5 micron repositioning, of the arm. But for the soft robot, it's silicone, that's more difficult. Yet, Paul, who was doing his PhD on this, could only manage to reach 1 millimeter precision in the positioning of this small silicone robot. So there is still room for improving this simulation. And the colleagues, they have different, I will not go too much in the engineering part, but they have different way, like closed, finite loop or open loop, to improve the simulation and try to achieve, our objective would be for this soft robot to be no more than 100 micrometer in accuracy of positioning.
And you mentioned at the end about there is a spin-off a company actually that is producing and selling this the spider mass. And one of the question was, what are the costs and also infrastructure that an hospital
Okay.
thinking about implementing this in a hospital will need to implement this type of technique methodology.
So the system is not so far commercial. The commercial version is under development because, as you can see, what we would like to target is something that would look like this, meaning something that's really like an echograph system that you can roll from one room, you do from another, that you can unplug. That is the mass spectrometer is on battery, so you can unplug in one room and the battery take. Keep the mass spectrometer, the vacuum, the time you move to the next room and plug it back. So, but that's not something impossible. So we are really discussing with Philip. So that is a CEO of the Cilios company to minimize the system and the cost of the mass spectrometer so that the price is still in line with what hospital or clinics can afford. However, we still target more decomprehensive cancer center or large clinic where they have maybe the possibility. I mean, they have enough money,
Sure.
to they can afford for these types of system. So we are really discussing because most of the mass spectrometers sold or which are actually on the market, they are including far too many things. Even if you take the Exploris, there are already far too many things for what we want to do. During
Sure.
the surgery, we will not perform the MSMS, we will not perform ETD, we will not perform ETD, HCD, whatever. So we are trying to see what we, get something that's really as small as possible. So far, we think that we could be able to sell a system that would be around 250,000. We hope we can reach 250,000 euros, which I think is still okay for a center, really a medical center that really need the system.
Thanks a lot. Thanks a lot. I have actually, thanks Justina also for this question. Justina, she's a co-chair also of the ETC. So I have actually a question, Isabelle. I was looking at the spectra you showed in the beginning, and you show very beautifully some multiply charged ions that were appearing. And I was wondering, did you see differences when you're acquiring positive and negative mode? Do you see more molecule appearing, like lipids appearing maybe in negative, protein more in positive? Does the instrument also that you use allows you to detect the different species?
So what is interesting with the spider bass is that because it's based on a concept, we have readapt a concept that's close to MALDI. We can definitively work both in positive and negative mode. I did not speak too much about that in the presentation, but definitively we can both work in positive and negative mode. From our experience for lipids and metabolites, both modes are useful. I mean, that's probably not of any surprise to you. For metabolites and lipids, you have lipids which are easier to ionize in the positive mode, some that are better seen in the negative mode, some that you see in both modes. So I would say that whenever we go for lipids, we always do D2. So we always try to record both the positive and the negative mass spectra.
I see.
And from what we have seen, it really depends. For some cancer, we have better classification. I mean, overall, we have similar performances in the two modes, but sometimes we have like 92% good accuracy of classification in the negative mode, 94 in the positive mode, or vice versa. And it depends the pathology. So which is logical because it also depends what are the upregulated lipids according to the pathology or the cancer. So yeah. So regarding protein, we are only working in the positive mode. I think we didn't really try in the negative mode, and we are working definitively in the positive ion mode. It could be interesting for us maybe to check in the negative mode, but we already try to improve in the positive mode, and that after maybe if we are more successful in the positive mode, we will shift to the negative mode. The difficulty that we have for protein is that it's not that the source cannot analyze protein, is that the protein are masked by the abundant metabolites and lipids, and think that even the protons are, you analyze more these molecules. And in multi-imaging, we have a solution, because in multi-imaging, what we generally perform when we do perform proteomics, imaging of proteins, whether they are intact proteins or proteins after trypsin digestion, is that before we perform tissue washing with organic solvent to remove the most abundant lipids of the tissue. So in multi-imaging, we can do so. In spider mass, if we want to apply this in vivo at one point, we will not be able to perform the delipidation of the tissue. So we are really trying to focus more on improving the analytical part, like using mobility, or maybe trying to change the parameter of the quadrupole so we can really cut the lipid range and only get past the protein signal in the orbit trap. So yeah, we are more working on the analytical side, but yeah, we know that we still have some progress. But yeah, one day maybe real-time proteomic will be true. Let's see. We still have some work.
Somewhere. I will ask Blandine if Blandine has some any question.
Yeah, I got some question regarding the first part of your presentation. You mentioned that you need at least 20 minutes or 50 minutes to get a pathologist to look at the tissue and give you a prognostic during the surgery. But for the same amount of time, what is the surface you can scan with the spider mask? Like, are you able to cover a larger surface in the same amount of time, or do you need to go through a small surface several times and so keep the same surface analyzed?
So to give you maybe a real example for ovarian cancer, in the intraoperative pathology, they can perform generally they go for three biopsy analysis
Okay.
and no more. And with if we could perform a continuous robotic scanning of the tissue as we did with the stiff robotic arm, we know that in 5 minutes now we can scan a rat bread, which is around 3 square centimeter. So
Okay.
I guess in the equivalent time, we've also a real-time result coming back from the interpretation with the machine learning or deep learning, whatever, with the artificial intelligence. I guess we can cover far more that they can nowadays perform. That's for sure.
Okay. Impressive.
I mean,
And so,
still have some development, don't mistake. We have very nice proof of concept ex vivo from histological tissue section, and we know we can calculate tabs and extrapolate. But our next goal is also to perform the first in man for oncology and see how much and fast we can apply that what is working in the lab inside the real environment.
and so talking about the real environment, what are the requirements you were facing to move towards the surgery block? Like, do you need to have a reproducibility check or instrument calibration monitoring that is crucial to have always the same measurements?
Yeah, I think that is also something that will be achieved by the spin-off company, looking to all
Okay.
these things. But what is planned is to have a system that not the surgeon, because the surgeons are not preparing the technologies, more the nurse that will do that, but that they will start switch on the system when arriving in the surgery in the morning, have a complete check of the system, including being able to check laser energy,
Yeah.
whether all the parts of the system are fully operating, whether calibration sounds, on track, and everything. And then only after this first, we would probably recommend to have maybe at least half an hour. So they start the system in advance to go for this complete checking, and then for sure that is mandatory. Then we know what is mandatory is that the system can be also submitted to the protecting, not leading to any contamination of the surgery room. So that means for sure the mass spec should be equipped with dry pump system, that for sure. Dry pumping system instead of oil vacuum pump. We will need also to replace the transfer tube for each patient, so to have something that we can unplug and replug, because we cannot avoid after a certain period of time that, we don't want to take the risk that some aerosol has condensed inside the tube, and that could cross-contaminate the analysis of the next patient. So we will just change the tube, and also we will need to be able that the optic fiber, all this part is within a gun of a protective gun, so to ensure that it's sterilized and there are no contamination possible inside the surgery room.
Yeah, the thing is that because you are a pioneer in the field bringing MS to the surgeon, I guess you have to establish your own quality controls and your own requirements to be sure of what is happening, I guess.
Yeah, I think for the INAV that was slightly easier because they are connecting to already existing surgical tools. So they already have the procedure for the knife or for the forceps. There is still the mass spec part, but there is already a part, the part that is in contact with the patient, for which they have a standard procedure. In our case, we will have to create all the procedures.
Yeah.
Celeos, Philippe perfectly knows that he will have a lot of time, effort, and money to give to regulatory aspect. Exchange a lot with different surgeons in different places, working on different cancers in different countries to be sure that he has in mind, everything it's needed that we will fulfill the requirements. Nevertheless, we will probably miss some things, that for sure. And the plans are to maybe get three first prototypes and install them in a cancer center in France in particular. So one probably in Lille, and at least one probably in Institut Gustave Roussy, that is quite famous in France, and probably one also in a hospital in Paris. And have a period where we will receive feedback from the end users, because we will probably have to re-optimize things. That's we already know. I guess that's how things are. I think it was the same for the development of the Da Vinci robot. They started with a first prototype. They release on the market, and they have since then they are constantly improving their robots. So.
Yeah. Thank you.
All right. So I think there are no other questions from the attendees. So I would like to thank you again, Professor Isabelle, for this wonderful talk. As Blandine said in the beginning, this has been recorded, so it will be available under the HUPO YouTube channels. I would like to thank, of course, Blandine and the HUPO office, Emma, also for all the support. Justina, co-chair of the ETC, is here, and I would like to invite everyone for the next talk, which will be on the 24th of April. But please visit the HUPO webpage for more information. Thanks a lot, and thanks to everyone.
Thank you.
Thank you so much, Blandine and Giuseppe, for this nice event.
Thank you for taking the time. Have a good one.
You too.
Bye-bye.
Bye.